Welcome, everyone. Thank you for joining us for today's webinar, Overcoming Challenges in EuCODE Cell Culture Platform Development, presented by Dr. Weimin Lin, Principal Scientist at Ambrx. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Lin. I will now turn the presentation over to her.
Good afternoon, everyone. First, I would like to thank the organizers for the invitation and the opportunities to share with you what I have learned about EuCODE technology and the cell culture platform development. The topic of my presentation today is overcoming the challenges in EuCODE cell culture platform development. Today, I will talk about what is EuCODE technology and how we overcome the challenges in EuCODE cell culture platform development. I will also provide a couple of case studies for each category, and then wrap up with some EuCODE technology applications and pipelines. So, what is EuCODE technology? U stands for eukaryotic. So, EuCODE is a unique engineered eukaryotic cell expression system. Here is a model of conventional expression system. Not like that. Ambrx, we engineered our own orthogonal tRNA and RS pierced into the cell lines to synthesize a protein consistent of non-natural amino acid at any site that can be used in conjugation without being interfered by the other 20 amino acids. The non-natural amino acid we're using is called PAF, which is also called the 21st amino acid. EuCODE technology has been implemented onto multiple platforms to create larger and more complex proteins where site-specific and homogeneous post-translational modification may be needed. For example, the EuCODE-enabled ADC. The Y-shaped atom is the antibody. The gray and the black atoms are the linkers and the payloads. The bright right sides are the non-natural amino sites. The EuCODE technology empowered us to be able to optimize the position of the conjugation, linker design, and the functional payloads. The key of the EuCODE technology is site-specific. Let me still use ADC as an example to show what advantages of having site-specific conjugation over the conventional conjugation. The conventional conjugation technologies lead to heterogeneous mixtures, which presents the challenges in optimizing the biological, physical, and the pharmaceutical properties of ADCs. The therapeutic windows or index for many ADCs that are currently in preclinical or clinical development remains very narrow. The site-specific conjugate ADCs have eliminated heterogeneity and demonstrated better CMC properties, such as generating homogeneous ADCs with few conjugated drugs, providing a more stable linkers in vivo. All of this can lead to wider therapeutic index. Everything is too good to be true, right? Here comes the challenges when generating the production cell line from the platform cell line. The non-natural amino acid, which is a PAF, is needed to generate a site-specific protein. Without PAF, cells can only produce truncated proteins. In the presence of PAF, cells can produce full-length proteins, but the suppression rate is only about 10 to 30%. It means how to improve productivity is going to be challenging down the road in process development. These are the step-by-step approaches from expression system development to clone selection and then process development. These are also the foundation of the EuCODE Cell Culture Platform. Cell culture process development starts with cell line generation and selection, followed by median and the process development and optimization in small-scale systems, including 96-well plates, shaker flags, and the bench-scale bioreactors for high-throughput screening purpose. Once conditions are defined, the process is often transferred to a pilot scale to test the scalability and produce materials for preclinical tox studies, and then large-scale manufacturing for production of a clinical material and the cGMP regulations. Once the development of a commercial cell culture process is completed at the lab and the pilot scales, the commercialization process begins with process characterization, scale-up, tech transfer, and validation of the manufacturing process. As cell culture technology is maturing, the biopharmaceutical industry has applied platform process to satisfy median demand and the quality requirement within a short period of time. The first step is to develop an expression system. Of course, you need to identify target and isolate gene before develop expression system. Therapeutic antibodies are mainly produced in mammalian host cell lines. The selection of the expression system is determined by its ability to deliver high productivity with acceptable product quality attributes and the preference of individual companies. A protocol needs to be set up to screen and select the highest producing and most stable clone. At this stage, the key is knowing genes for the protein you want, what cell line to use, and what clone form that cell line is the best. After cell line, there's perhaps no other factor like a culture medium that can more greatly influence the process performance. Once a successful growth and the production medium for each cell line has been developed, the culture medium formula and the details of how it was developed are often two of the most closely guarded secrets of the manufacturer. It indicates how important medium development is to overall process development. So many commercial mediums are ready to use. Hydrolysis is so powerful. Do we still want to develop chemical-defined medium? How can we identify the components in culture medium? How many combinations? When to feed them? How about inducers or promoters? These are the questions we need to answer one by one. Nowadays, using chemically defined medium have become the standard in the biotech industry. Here are some of the motivations of why we need to develop chemical defined medium. These motivations are pretty much well known now, so I won't read through all of this. But next, I would like to share some experimental strategies. Several approaches can be used systematically, such as single component titration, then medium analysis, and the medium blending. Cell line is very sensitive for the medium change. So normally, you should start with all needed nutrients using published formulation. You can group all nutrients based on the solubility or the nutrient category, and then try dosing response titration and DOE studies. Here is the preferred workflow. At the very bottom, you start with the single component screening, then go up to the screening model, then you adjust your formulation, and then testing, then measure your uptake rate and performance. After several rounds, eventually you will success. Those dose response titration studies are very time and labor consuming. So you can modify your shake flask screening protocol. Instead of taking 14 days of a full production time, you can initiate the production stage in a larger vessel for 6 or 7 days, and then split into shaker flask with different conditions or factors. After 24 to 72 hours, measure VC and titer. If there's no positive response, do nothing. If there's a showing positive response, moving forward to DOE study. If you don't have Amber 15, buy it and you're gonna love it. In this study, we try to replace a hydrolysate containing commercial media with an in-house developed chemical defined medium. Initial basal medium derived from IMDM with enrichment in amino acids and the trace elements. Initial feed medium was partially concentrated from basal medium with the removal of excess inorganic salts. The amino acid concentration was determined by calculation of uptake rate. Titration of a trace element and the supplementation of vitamins in both basal medium and the feed medium, you can see the graph. The commercial medium is in dark blue, and the chemically defined medium is in light blue. Comparing the growth profile, VCD is much higher in commercial media. Viability is about the same. It is interesting to see that even VCD in chemical defined medium is lower than in commercial media. The harvest titers are very similar. It means the QP in chemically defined media is much higher compared to in commercial media. So the enhancement of a specific productivity per cell is achieved by combining both optimization of a medium composition and the bioreactor operation conditions. PQ here on the right is show very comparable. So those results tell us it's not unusual to see this kind of improvement. Since you are the only one, know your cell line the most, not the vendor. If it's possible, you should develop your own medium. Of course, chemical-defined medium is preferred. Normally, process development is often divided into an early and a late stage, with each having different goals. The goal of early stage development is to rapidly develop bioprocess to produce materials for phase 1 and phase 2 clinical trials and tox studies. Standardized process conditions and procedures are essential. The process may not be fully optimized for every molecule. As the product moves into late-stage development, the process can be further optimized. To optimize the conditions for biomanufacturing process normally is in a scaled-down version, like bench scale, but some companies even use Ambrx 15 for process development for early stage, and then directly scale up to 2,000 liters. When you optimize the process, you need to consider what temperature, any shifts, what oxygen level, CO2, pH, any shift, when to harvest. When we were working on the process optimization, we all need to remember that the most important thing is to deliver meaningful, scalable data. Culture operating parameters include physical, chemical, and biological parameters. Physical parameters include temperature, gas flow, and agitation speed, while the chemical parameters include DO, CO2, pH, osmolarity, and metabolite levels, including substrate, amino acid, and waste byproducts. Biological parameters are usually used for determining the physiological state of the culture and including VCD, viability, and more the others. Variations in the macro environment parameters from optimal levels can have a dramatic impact on the culture performance, productivity, and the product quality. Appropriate online and offline sensors can provide a lot of information that can enhance the process control. I was supposed to share a temperature shift modeling case study, but rejected by the legal. So I will share a feeding strategy optimization case study. After platform process established, optimizing a feeding strategy is super critical to achieve a culture that both grows and produces protein effectively. In this case study, DOE-based process optimization on feeding strategy of nutrient, inducer, and growth factors were done. A simplified feeding strategy was implemented during the clone selection, and the titer was increased twofold. A more complex feeding strategy was implemented during the process development, and the titer was increased 5 to 6-fold. So cell line, medium, and the feeding strategy are three key for the cell culture platform development. The ultimate goal of all the work we have done so far is for us to be able to scale up the process in large-scale bioreactor for production of therapeutics. During bioreactor operating condition scale-up, agitation and aeration are two important conditions to be scaled up properly to achieve comparable process performance across different scales. Agitation is adjusted to provide sufficient mixing and oxygen mass transfer, and is generally scaled using eco-specific energy dissipation rate whereas aeration is modulated for appropriate supply of oxygen and the removal of excess CO2. In small-scale bioreactors, the operating parameters are typically wider because mixing is generally not a problem even at relatively low agitation speed, and the CO2 removal is efficient due to large surface to volume ratio. In large-scale bioreactors, these parameters need to be selected carefully as agitation-related shear force, for example, impeller tip speed, and aeration-related CO2 accumulation may increase with skill. A qualified cell culture scale-down model is very crucial. It can provide the necessary information to assess and resolve any unexpected performance problem of the large-scale system. Beyond manufacturing support, the scale-down model can also be used for process characterization and validation experiment to study acceptable ranges of process operating parameters and determine the critical operating parameters. Anti-HER2 is our first clinical program. In this case study, we successfully scale up from 5 liter to 500 liter and then 2,000 liter. We build up the scale-down model, and then later we implement anti-HER2 scale-down model to develop anti-PSMA program. Therefore, later, of course, when we scale up anti-PSMA program, there was no issues from 5 liter to GMP 500 liter. So this is indicate scale-down model is super useful for you doing the scale up for any process. We have developed EuCODE Cell Culture Platform. The system can rapidly provide the amount of material needed at all stages of study for antibody molecule selection, ranged from milligrams of screening needs in 2 weeks to 100 grams of tox run in 8 weeks. Once lead molecule is selected, stable cell line can be generated in 6 months with titer about 1 to 2 grams per liter to support clinical trials and potential commercialization. In summary, EuCODE Cell Culture Platform has been developed and validated by multiple programs. The productivity of a platform cell line was improved by 5 to 4 to about 5 to 6 fold. The production cell line generation process was shortened to 6 months by using platform cell line engineering and the process optimization. And the multi-programs have been demonstrated titer of 1 to 2 grams per liter of manufacturing ability and up to 2,000 liter of scalability. Because the established EuCODE Cell Culture Platform can generate site-specific and homogeneous biomolecules, it empowers EuCODE technology to many applications. Besides ADC, we also have half-life extension, bispecific, CAR-T, vaccine, and more in drug development. EuCODE technology is the best-in-class non-natural amino acid technology. There are many pipelines currently in clinical research. If you are interested in EuCODE technology or EuCODE Cell Culture Platform, please contact us. This is definitely a team effort between the cell line development group and the cell culture development group. Thank you so much for your attention.
Thank you, Dr. Lin, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. And before we go, I want to let everyone know that this webcast will be available for on-demand viewing through April of 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you next time. Goodbye.